Hi everybody, today is all about the chromosomes. We're going to zoom into and see what really makes up the chromosome structure that we're familiar with. The chromosome that we're going to look into is from the domain Eukarya. The members of the Eukarya domain, their chromosomes tend to be much more complex than other domain organisms, such as bacteria or viruses. They're more complex and intricate in that they have substantial amount of protein associated with the chromosomal DNA in all phases of the cell cycle. So here we have an example of a human chromosome. This chromosome appears to be quite condensed. When a chromosome is this condensed and appears to be in this structure and morphology, it tells us that it must be going through some type of subdivision, either mitosis or meiosis. Because after the chromosome separation and the cell division, the cell enters another phase of the cell cycle. The cell enters the phase called interphase, at which time the components of the chromosome uncoil and decondense into a form referred to as chromatin. And the chromatins are going to be dispersed throughout the nucleus during this interphase. A chromatin is basically a complex of DNA and protein. By the way, this is a metaphase chromosome. And remember from previous videos that this particular part of the chromosome is known as chromatid. Now let's zoom in closer. So again, when a chromosome starts to uncoil and decondense, we get these fiber-looking structures. They're called chromatin or chromatin fibers. Going further down and zooming into the chromatin fiber, this is when we start seeing the complexity of the DNA organization that makes up the chromosome and the chromosome structure. We have these multiple entities that are organized in a very specific way. This is a protein called H1 histone. And this part of the chromatin fiber is the nucleosome core. And together, this organization is known as solenoid, and it's measured to be around 30 nanometers in diameter. In addition, this part of the chromatin fiber, when you see it's twisting and turning like that, we refer to them as looped domains. Let's move even further down. Here we have more of the histone proteins. If we look closely into the core of the nucleosome, we're going to find this complex that consists of 8 proteins, and they're being wrapped around by the strand of DNA. This complex is a combination of H2A, H2B, H3, and H4 histone proteins. So there are 2 of each, and we refer to them collectively as a histone octamer. You might wonder what the functions of these histones are. Well, as a unit, one of their main functions is to package the DNA into structural units called nucleosomes that we have already observed. I can label them here, nucleosomes. There are 2 of them. The histone octamer also plays a role in gene regulations. For example, the transcription of the DNA surrounding them here in pink. This part here is the H1 histone protein that we've also seen before, and it has its own place between the 2 nucleosomes unopaque space here, which we call the spacer DNA. Or linker DNA. Finally, we've arrived at the smallest functional entities group of the chromosome composition here. Guess what? It's DNA. We see a double-stranded DNA here, and it's about 2 nanometers in diameter. Of course, we can zoom in even more further to see the different enzymes and even smaller proteins that work together on a DNA strand. But for now, we're just going to stop here. So we've learned together that when a cell leaves the cell division, the chromosomes are going to be broken down into smaller structures called chromatins. But these chromatins will start to coil and condense back into visible chromosomes again when they need to re-enter the cell division.